The next topic to review is the all-important topic called kinetics. I realize that most medical students despise this topic of kinetics, and the reason why I think most students don't like kinetics is because there's a lot of mathematics, there's a lot of equations, and sometimes it's not taught properly. But in this topic of kinetics that we're going to be reviewing, there's going to be no mathematics. It's all going to be conceptually driven. But it's a matter of understanding the basics of kinetics. You must understand kinetics to understand pharmacology, to understand what goes on in pathology, because it's the basis of diseases, it's the basis of pharmacology, is kinetics. So what we're going to be doing in this review of kinetics, I'm going to try to simplify as best as I can and talk about it from a concept point of view and minimize any type of mathematics and equations. Now, in terms of the context of concepts regarding kinetics, why do you have enzymes in the first place? And the reason is what enzymes are doing is they're trying to make reactions go faster. Because in the body, reactions can take place if a substrate making a product. If the delta G is less than 0, it means it will happen. If the delta G of a reaction is less than 0, it means it's spontaneous, it will happen. But it might take a day, it might take a week to take a substrate to make a product. It's too slow. And so therefore you have an enzyme encoded by a gene in a chromosome. And the enzyme makes the reaction go within seconds. So the idea here on this table on the left side, when a delta G is less than 0, it means it will happen, but it might be too slow by itself. When a delta G is greater than 0 of a substrate making a product, that means it will never happen unless energy is supplied. When a delta G is equal to 0, it means it's in equilibrium. When a delta G 0, that's under standardized conditions, that's not that important. The important feature is when a delta G is less than 0, it means it will happen. But it's not saying how long it takes. And why you have an enzyme on the right side of this table is when an enzyme does, it makes the reaction go faster. Instead of taking a day or a week, the reaction may take place within seconds. That's what an enzyme does. Now, if the National Board were to ask you, what does an enzyme do in an enzyme catalyzed reaction to make the reaction go faster? What you say is what an enzyme does. It decreases the energy of activation called delta G star. All right, now we need to review. What does this mean? Delta G star versus delta G. These are two issues of energy. What's the difference? What delta G is the inherent energy of a substrate. Every substrate has energy. Fatty acids have energy, glucose has energy, amino acids have energy. So the inherent energy of a substrate is called delta G. Now, for a substrate to give up its energy, the substrate must first acquire a little bit of energy from the environment. And the energy from the environment is called delta G star. In the atmosphere right now, in the room that you're in right now, there's energy in this room. That's called delta G star. The energy from your body temperature, the energy from the lights, the energy from the computer, the energy in the environment is called delta G star. So for a substrate to give up its inherent energy called delta G, the substrate must first acquire a little bit of energy from the environment called delta G star. Once the substrate acquires the delta G star energy from the environment, then the substrate gives the delta G star energy back to the environment and then gives up and releases its own energy called delta G. And what an enzyme does, as simple as I can state, is the enzyme decreases the delta G star energy from the environment a substrate must acquire. An enzyme decreases the amount of delta G star energy from the environment a substrate must acquire. And by decreasing the delta G star, therefore the reaction takes place faster. Now there's a diagram that could illustrate this on the next figure. In this graph is a diagram showing what I just mentioned in words a few minutes ago, where a substrate to form a product must go through a transition state called S star, meaning the substrate has more energy in it than the substrate itself. The substrate has energy called delta G, and this product also has energy, but S star has more energy because it acquires energy from the environment called delta G star. So here is the energy from that the substrate has, called delta G, and then it's getting energy from the environment, and that's called delta G star. And so now that substrate has extra energy in it. Eventually that substrate, once it acquires the energy from the environment, the substrate will give up its energy from the environment back to the environment. That's called delta G star. And eventually it gives up its own energy called delta G. So the idea here is that delta G from the environment, delta G star from the environment, that determines the rate. Because what an enzyme does, it lowers how much energy from the environment the substrate must acquire. And by lowering this difference right here, determines the rate. The less energy delta G star from the environment, the less energy the substrate must acquire, the faster the reaction can take place. Notice whether the enzyme is present or not. That is independent of the enzyme. This part here, delta G, is not affected by the enzyme. So the enzyme, what it's doing is it affects delta G star, not delta G. So one more time, what an enzyme does, it lowers how much energy from the environment a substrate must acquire, called delta G star. And once it acquires it, the enzyme lowers that amount. By lowering it, the reaction goes faster. And delta G is not affected by the enzyme. So on board exams, if they were to ask you, what does an enzyme do in an enzyme catalyzed reaction? Never ever state an enzyme affects delta G, the inherent energy of a substrate. It doesn't. What an enzyme does, it affects the delta G star, the energy from the environment. By lowering that amount, the reaction goes faster. Coming up next are back-to-back graphs. And one's a Michaelis-Menten plot. The next one after this is the Lineweaver-Burk plot. Now in the Michaelis-Menten plot, there is an equation called the Michaelis-Menten equation. Here is the Michaelis-Menten equation for board exams. Not important. Let me emphasize. There's no mathematics that's going to be high yield for board exams regarding the Michaelis-Menten equation. So this Michaelis-Menten equation, I'm going to eliminate it. Watch it disappear. Gone. Don't worry about it for board exams. You might see it in other books, but for boards, it is low yield. What you must do, though, what's high yield for board exams, is interpretation of a Michaelis-Menten plot. On a Michaelis-Menten plot, where do you find two major terms? One term is called the Vmax, the other term is called the Km. And what a Vmax is, by definition, it's the maximum velocity, how fast a reaction can take place. The ability of an enzyme to carry out the conversion of a substrate to a product. So on a Michaelis-Menten plot, where do you find the Vmax on the graph? You find it where the graph, the hyperbolic line, reaches a plateau. That is the Vmax. So in this example, the Vmax is set at a value of 50 micromoles per second in this example. So this is the maximum velocity at which an enzyme can carry out the substrate to form a product. And what's rate limiting is the enzyme. The enzyme is working as fast as it can. It's working at its maximum velocity. It's working as fast as it can. Vmax. Now the Km value, what's found, where you find it, the Km is found at 1/2 of the Vmax. If you were asked, what is the definition of a Km? You answer by saying a Km is a substrate concentration, meaning it's going to have units like millimoles, milligrams per deciliter, A Km is a substrate concentration at which the enzyme is working at 1/2 the Vmax. So if you know where you find the Vmax, right here, 1/2 of that is 25. So at that value of 25, then you go to the enzyme line, and then straight down, that's the Km. So in this example, the Km is at 2 millimolar, meaning at 2 millimolar, the enzyme is working at 1/2 of its ability, 1/2 of the Vmax, on a Michaelis-Menten plot. The next graph to talk about is the Lineweaver-Burke plot. Now, pharmacologists tend to use the Lineweaver-Burke plot compared to a saturation plot. But in a Lineweaver-Burke plot, there is an equation called the Lineweaver-Burke equation. You see it here, the Lineweaver-Burke equation. But just like the Michaelis-Menten equation, for board exams, low yield. So I'm going to eliminate it, even though you might see it in textbooks and other review books. But for boards, it's not important. They're not going to ask you to use the Lineweaver-Burke equation. What's critical, though, is to interpret a Lineweaver-Burke plot. Now, a Lineweaver-Burke plot is a double reciprocal plot. What you need to know about a Lineweaver-Burke plot are two things. Where do you find a Vmax and where do you find a Km? Now, where you find a Vmax on a Lineweaver-Burke plot, you find it at the intersection of the y-axis and the enzyme line is where you find the Vmax. And remember, because it's double reciprocal, that value found at the intersection of the y-axis and the enzyme line is 1 over the Vmax. Not the Vmax, it's 1 over the Vmax because it's a double reciprocal plot. Where do you find a Km? On a Lineweaver-Burke plot, it's found at the intersection of the x-axis and the enzyme line. And that value is minus 1 over the Km. Now, please don't get misinformed here. Remember, a Km is a substrate concentration. So on a board exams, if they show you a Lineweaver-Burke plot, they will say 1 over the substrate concentration for the x-axis. And because a Km is a substrate concentration, the Km will be 1 over the Km. And because it's a negative value, a negative times a negative, in this example here, minus 0.5, and it's a minus of that. So therefore, a negative times a negative is a positive. So don't get bogged down on that is minus 1 over the Km. But just note that it is minus 1 over the Km. So one last thing, just to summarize. On board exams, what you need to know about a Lineweaver-Burke plot are two things. Where do you find the Vmax and where do you find the Km? Now, why is that important to know where you find the Km and where do you find the Vmax? Because of all important integration with pharmacology on the next figure. Here comes the integration of biochemistry with a lot of pharmacology and a class of drugs that are used in pharmacology. In pharmacology, the drugs, generally speaking, are classified as one of two kinds, competitive or non-competitive drugs. Biochemists tend not to use the word antagonists or agonists compared to pharmacology, but the idea is these are inhibitors, whether they're competitive inhibitors or non-competitive inhibitors. So competitive inhibitors resemble the substrate. Competitive inhibitors or competitive drugs, they look like the substrate structurally, and they compete to get into the active site of the enzyme. So competitive drugs look like the substrate. They compete to get into the active site of the enzyme. So those are competitive inhibitors or competitive drugs. Non-competitive inhibitors, they don't look like the substrate, and they bind to the enzyme somewhere else on the enzyme. They do not get into the active site, or the non-competitive drugs, non-competitive inhibitors. They don't look like the substrate, they don't resemble the substrate, but they bind somewhere else on the enzyme, regulatory sites on the enzyme. Now, what you need to know on this table is competitive inhibitors, what they do is they increase the Km value, but they have no effect on the Vmax. Competitive inhibitors do not affect the velocity of the enzyme, the Vmax of the enzyme. but they will affect the Km of the enzyme. Whereas non-competitive inhibitors, they have no effect on the Km, but they decrease the velocity. Because if a non-competitive inhibitor binds to a regulatory site, it prevents the enzyme from undergoing a conformational change properly. The enzyme won't work, and the enzyme will not be able to convert a substrate to a product efficiently. That's why the Vmax decreases when a non-competitive inhibitor binds to an enzyme. So now regarding competitive inhibitors, many drugs are competitive inhibitors. In fact, most drugs, if they were to ask you, if a drug is it a competitive or non-competitive inhibitor, if you don't know the answer, take a guess and say competitive, because most drugs are competitive inhibitors of enzymes. Notice here in this example, show you two examples of competitive drugs, all statin drugs. There are 6 statin drugs that are commercially available to treat hypercholesterolemia. All statin drugs are competitive inhibitors of the substrate HMG-CoA for the enzyme HMG-CoA reductase. Another example of a competitive inhibitor, methotrexate, is a competitive inhibitor of dihydrofolate reductase because methotrexate looks like it competes with dihydrofolate, the substrate for the enzyme dihydrofolate reductase. So most drugs are competitive. There are some non-competitive inhibitors of enzymes. A great example of this is allopurinol for the enzyme xanthine oxidase, a non-competitive inhibitor of xanthine oxidase. But most drugs are competitive. Now, one way they can ask you about competitive or non-competitive inhibitors of enzymes in pharmacology as well as in biochemistry, they can show you a Lineweaver-Burke plot. Here we have an enzyme and some drug is being added to the enzyme. And then he can ask you, is this drug that's being added to the enzyme, is it non-competitive or is it a competitive inhibitor of the enzyme? And the way you do this with a Lineweaver-Burke plot is, what they might do is, they might say to you, this is a Lineweaver-Burke plot of an enzyme all by itself. And this is an enzyme when some drug or inhibitor is added. What type of a drug is it? So the way you do this with a graph on the left side, you say to yourself, where do you find a Vmax? You find it at the intersection of the y-axis and enzyme line. Did the Vmax change when the drug was added? And the answer is no change. Where do you find the Km? It's found at the intersection of the x-axis and the enzyme line. Did the drug change the Km? And the answer is yes. The Km changed. The Vmax no change, so you say what's being added to the enzyme is a competitive inhibitor of the enzyme. There's competitive inhibition. On the right graph, they say to you, this is an enzyme all by itself, and this is the same enzyme when some drug is added. What type of drug is it? You say to yourself, where do you find the Vmax? It's found at the intersection of the y-axis and the enzyme line. Did the Vmax change when the drug was added? And the answer is yes in this example. Where do you find the Km? It's found at the intersection of the x-axis and the enzyme line. Did the Km change? And the answer is no change. And so because the Km no change, the Vmax decreased. That means what's being added to the enzyme is a non-competitive inhibitor. So they can show you a Lineweaver-Burke plot where some enzyme is present and some drug is added, and they ask, is it a competitive or non-competitive inhibitor? All you got to do to yourself is ask, where do you find the Km? Where do you find the Vmax? Did they change or not change? And you get to the answer. Is it a competitive or non-competitive drug or inhibitor being added to the enzyme? Now, there's another situation that might occur in which you see a Lineweaver-Burke plot that has nothing to do with pharmacology, but has a lot to do with endocrinology. Because what you might see on board exams is a Lineweaver-Burke plot where you have an enzyme all by itself. And then they can say to you on the board exam that some hormone was being secreted. And the hormone then changed the Lineweaver-Burke plot to now have the line of the enzyme look like this, as shown by the arrow. And so what's happening here is that Vmax is changing. and the Km stays the same. And so what's happening here is when you secrete a hormone and it changes the number of enzymes, there's more enzyme around. Or when some activator is present, the velocity is increasing, the Km no change. That means some hormone is being released to increase the number of enzymes. So one more time. If the velocity is increasing and the Km is not changing, what that's telling you is either a hormone is being released to increase the number of enzymes, or an activator is being generated to increase the activity of a particular enzyme. Because the velocity is increasing, the Km stays the same. Also, another issue here. Please don't get confused. On the y-axis, when going from top to bottom, you're increasing velocity, not decreasing velocity. Don't be fooled. Without the more enzyme, we're at the top of that line, but now when more enzyme is around, the velocity now is increased, not decreased. Don't be fooled. The velocity is increasing because it's a double reciprocal plot, but the Km stays the same. Here is a very important bridge to pharmacology, and that is methanol poisoning, also called wood alcohol. Wood alcohol can kill human beings, and if the boards were to ask you, how does methanol kill? And you answer by saying what happens is methanol undergoes the same metabolism as ethanol. Methanol sees the enzyme alcohol dehydrogenase and converts it to formaldehyde, which then sees the enzyme acetaldehyde dehydrogenase, which converts it to formic acid. So methanol generates formaldehyde, then formic acid. And formaldehyde, as well as formic acid, can leave the liver where the metabolism occurs. These can enter the bloodstream, they can go to the brain, damage the brain, and can kill human beings. And so what is the antidote for methanol poisoning? And that is ethanol. Because ethanol is a competitive inhibitor of alcohol dehydrogenase. And so if you give ethanol to somebody who's methanol poisoned, ethanol will now compete with methanol for the enzyme alcohol dehydrogenase, preventing methanol to convert to formaldehyde. And methanol itself is non-toxic. It'd be dumped into the blood and be excreted into the urine, not causing problems. So why you give ethanol is to prevent alcohol dehydrogenase from converting methanol to formaldehyde. If the boards were to ask you another issue of methanol poisoning, they might ask you what is in pharmacology, what drug can be used to treat methanol poisoning. Ethanol is not really a drug, but there is a drug that's commonly used to treat methanol poisoning, and that's fomepizole. Fomepizole is a competitive inhibitor of methanol poisoning. It prevents alcohol dehydrogenase from converting methanol to formaldehyde. That's fomepizole. Another item about kinetics. Everything that was mentioned earlier regarding enzyme kinetics, Kms, Vmaxes, competitive inhibitors, non-competitive inhibitors, Lineweaver-Burke equations, and so on, everything about enzyme kinetics also is applicable to transport kinetics. Transporters have Kms, they have Vmaxes. There's things like Michaelis-Menten plots and so on, Lineweaver-Burke plots. Everything is identical. So we're not going to go through an exhaustive review of transport kinetics because it's the same thing as enzyme kinetics. But as an example of transport kinetics are the GLUT transporters. In this example here is a graph, a saturation plot of GLUT1 versus GLUT2 versus simple diffusion. Why do you need transporters for glucose, as well as most polar substances? Now, in the white line is simple diffusion. Because glucose is a polar molecule, it cannot go through a membrane. And you need to get glucose into cells in order to metabolize to make ATP. If there were no transporters around, glucose would hardly enter a cell. You wouldn't make ATP, and you would not be able to sustain life. So you need a transporter. And there are two major kinds of transporters for glucose. One is GLUT1, the other is GLUT2. And the big difference between the two, GLUT1 and GLUT2, besides location. In physiology, they talk about the locations of these two transporters, but in terms of the biochemistry and the pharmacology of these two transporters and the physiology of these transporters is, besides location, they have different Km values. GLUT2 has a very high Km value. It's a very high Km value, meaning first order kinetics. Meaning the more glucose in the blood, the more glucose enters cells. GLUT2 is found, as an example, in the liver. And because GLUT2 is located in the liver, the more glucose in the bloodstream, the more glucose enters the liver by first order kinetics. GLUT2 never saturates because you can never eat enough glucose to be at the Vmax of GLUT2. Notice that blue line? It's going up as the glucose concentration on the x-axis goes from 5 millimolar, which is normal, to 10 millimolar to 15 millimolar and greater. That blue line, the dark blue line, never saturates. You never attain Vmax with GLUT2. Whereas GLUT1 has a very low Km value, that light blue line. Notice here, below 5 millimolar, the more glucose in the blood, it starts to rise and it reaches plateau at 5 to 6 to 7 millimolar glucose. So the more glucose in the blood, the GLUT1 transporter, because it has a very low Km value, will reach saturation, will reach the Vmax at about 6 or 7 millimolar glucose. So what that means is the more glucose in the blood, the GLUT1 transporter will attain zero order kinetics. The more glucose in the blood, you're not going to transport any more glucose into a cell. GLUT1, as you know in physiology, is found in most cells in the body, skeletal muscle, for example, but most commonly it's found in brain, erythrocytes, and so on. At 5 millimolar, which is normal blood glucose, look what's happening. In 5 millimolar glucose, hardly any glucose enters the cell by simple diffusion. Hardly any glucose enters the liver because the liver has a GLUT2 transporter. Most of the glucose is entering at 5 millimolar, is entering cells which have the GLUT1 transporter. And that is the brain, erythrocytes, and most cells in the body at 5 millimolar. The only time that glucose would enter the liver in appreciable amounts is when you eat a high carbohydrate meal. When you eat a high carbohydrate meal, the blood glucose rises from 5 to 10 millimolar to 15 millimolar. Notice cells which have the GLUT1 transporter are at Vmax. No more glucose entering those cells. Zero order kinetics. But cells which have the GLUT2 transporter, like the liver, the more glucose in the blood, the more glucose enters those cells because it's first order kinetics. And in those cells, the more glucose enters the liver will take that glucose and convert the glucose to fatty acids, making triglycerides, making cholesterol, making glycogen for storage. Another situation in which saturation kinetics might be found is with allosteric enzymes. Now, what an allosteric enzyme is, it's an enzyme that has a sigmoidal plot to it. Here is a control. It's an enzyme all by itself. It looks sigmoidal, where S-shaped curve, and then that's telling you it's an allosteric enzyme. And every pathway in the human body, every metabolic pathway, whether it's glycolysis, whether it's fatty acid synthesis, or whatever, every pathway has at least one regulated enzyme. It's an S-shaped sigmoidal curve. Now, what's important about an allosteric enzyme is if an activator is added to the enzyme, the activator will shift the curve to the left. So here's an activator that now will shift the curve to the left, making the enzyme work better. If, on the other hand, an inhibitor is added to the enzyme, it'll shift the curve to the right. Now, biochemists tend to use the word activators. Pharmacologists tend to use the word agonists, but it's one and the same. Pharmacologists tend to use the word antagonists. Biochemists tend to use the word inhibitor. It's one and the same. But the idea is if an antagonist is added to the enzyme, the curve will shift to the right, slowing down the enzyme from working. What's coming up next is a series of 7 questions that come from the end of the chapter. My advice is pause the recording and do these 7 questions on your own first. After you do these 7 questions on your own, then come back to the recording and listen to the explanation of these questions. So we're going to do questions 1 through 4 and questions 6 through 8 at the end of the chapter. Question 1. It says there's a peptide of 5 amino acids. It's treated with peptidase to generate 5 individual amino acids. The individual amino acids are then subjected to electrophoresis. At the top is the negative terminus. At the bottom is the positive terminus. At the dotted line, if the mixture of the 5 amino acids are placed at the dotted line, you see a variety of spots. And they're asking in this vignette, the amino acid indicated by the arrow is which of the 5 amino acids? And the answer is option B, arginine. And why is arginine the answer? Because arginine, as we mentioned earlier in this chapter, it's the most strongly positively charged amino acid. It migrates quickest to the negative terminus. If the National Board were to ask you a vignette like this, and they ask you, what is the amino acid on this paper electrophoresis? That is one shown by this arrow. And the answer is that particular amino acid would be glutamate. If they were to ask you what amino acids do not migrate that are at the dotted line, there are 2 amino acids of the 5. That would be either glycine or alanine, because the R group, they are non-charged amino acids. Glycine and alanine, remember they have aliphatic side chains as the R group. The last amino acid of the 5 migrates to the negative terminus, but not nearly as fast as arginine. If I were to ask you which one is this amino acid, and the answer is that's histidine, because histidine is weakly positively charged. But it will migrate to the negative terminus, but not nearly as fast as arginine. The next question deals with an enzyme that's discussed in Chapter 12, phosphofructokinase 1. This is an enzyme, it's the main regulated enzyme of glycolysis. And it says in this particular question, it has a delta G value of minus 3.5 kilocalories per mole. What does that number mean? And the answer to this question is option G. It cannot be used for gluconeogenesis. This enzyme has a negative value of the delta G minus 3.5. Yes, it is. It means it's irreversible. And so the answer is not A, which states reversible. It has nothing to do with time. Option B occurs very slowly. Remember, the delta G has nothing to do with time. That value has nothing to do with activators, inhibitors, has nothing to do with energy of activation. Inhibited by ATP. Option D says inhibited by ATP. Yes, it is inhibited by ATP, but the value has nothing to do with inhibitors or activators. What the value means, it is irreversible. And because it's irreversible, it cannot be used for gluconeogenesis, which is the reversible pathway compared to glycolysis. Gluconeogenesis is the reverse of glycolysis. Glycolysis is where you take glucose and you burn it. Gluconeogenesis is where you take carbon and you make glucose. And because these are reciprocal pathways, this is an enzyme which has a negative value of minus 3.5. It means gluconeogenesis cannot use this enzyme. There's another enzyme in gluconeogenesis that takes the product and makes it into the substrate, and that is an enzyme called fructose 1,6-bisphosphatase in gluconeogenesis, not glycolysis. The next question looks formidable because what it is a series of substrate values, a series of velocity values, and it looks like it's going to entail a lot of mathematics. But in fact, there is no mathematics here. It's simply a conceptual question. Simply just look at the values and picture what's going on, and you can easily determine the answer. The answer to this question is option E. 0.5 millimolar is the Km value. Now, how do we get that value? Because the Km is equal to the substrate concentration at which the enzyme is working at 1/2 the Vmax. So the idea here is to get to the answer, look at the velocity values and look at the substrate concentration. Starting at 5 millimolar substrate, up to 10, up to 50, up to 100 millimolar. As you increase the substrate concentration, notice the velocity is not increasing very much. At 5 millimolar, the velocity is 91, 10 millimolar, the velocity is 95, and so on. And because the velocity is not increasing, that means you're at the Vmax. The Vmax, therefore, is 100. And because a Km is equal to the substrate concentration at which the velocity is 1/2 the Vmax, that means 1/2 of 100 is 50. You go straight across from 50 and you find out it's 0.5. So that's why the answer is option E, 0.5 millimolar. So it's not a difficult question if you're thinking about what is the definition of Km. The next is a series of saturation plots, and they're coming back to the same enzyme called phosphofructokinase 1, the key regulated enzyme of glycolysis. And here they're saying, what is the effect of ATP on the enzyme PFK1? And what's important to note is PFK1 is inhibited by ATP. So ATP is an inhibitor of the enzyme. It's an allosteric enzyme affected by an allosteric inhibitor, ATP. So what does ATP do? Because it's a competitive inhibitor of the enzyme, it will shift the curve to the right. So if the solid black line is ATP enzyme phosphofructokinase 1, remember, allosteric enzymes gives rise to a sigmoidal curve, an S-shaped curve, and because it gives rise to an S-shaped curve, we can eliminate option D because that's a straight line. Option E is a hyperbolic curve. Option C also is hyperbolic curve. So we can eliminate option C, D, and E because they are not S-shaped sigmoidal curves. Option C, D, and E. So the answer will either be option A or B because the enzyme all by itself is sigmoidal. And so because ATP is an inhibitor of the enzyme, it shifts the curve to the right. That's why the answer is B. ATP plus ATP shifts the curve to the right. Whereas on option A, the curve shifts to the left in the presence of ATP. And that would be if there's an activator of an enzyme, and ATP is an inhibitor, not an activator. This last question deals with a seemingly difficult series of 3 questions. But in fact, again, these 3 questions are somewhat similar as long as you understand what a Km value and what a Vmax value are all about. There is no mathematics in any of these 3 questions. So what in this vignette, what they're doing is they're giving you 2 enzymes, enzyme A and enzyme B, and they're giving you Km values for both of these enzymes and Vmax values for these 2 enzymes. And in question 6, they're talking about one of the 2 enzymes, enzyme A. In enzyme A, they're saying at a concentration of 5 × 10⁻⁶ molar substrate, the velocity of the reaction catalyzed by enzyme A will be, and the answer is A. Now the best way to figure out the answer is in your mind, or I'll show you, in your mind, just draw a generic saturation curve. Here is a generic saturation curve. Now remember, on the x-axis is the substrate concentration, on the y-axis is velocity. And it says enzyme A, the Vmax is 20 for enzyme A. So we can put, because we know where we find the Vmax, we can put that value of 20 for the Vmax. And where do you find the Km? The Km is found at 1/2 the Vmax. So 1/2 of 20 is 10. And then 10 will be found right here. So this is the value of 10. You go to the enzyme line and straight down. That value there, it says, is 5 × 10⁻⁶. So let's put it down. 5 × 10⁻⁶, the enzyme is working at 1/2 the Vmax. So now they're asking, at a concentration of 5 times 10 to the minus 6 molar, which happens to be the Km. So at that substrate concentration of 5 times 10 to the minus 6, what is the velocity? And the answer is 10, because we're at the Km, at the substrate concentration of 5 times 10 to the minus 6. What is the velocity? The answer is 10. Question number 7. Now we're talking about enzyme B, and with enzyme B, the answer will be option B, 15, enzyme B. Now, it says a concentration of 5 times 10 to the minus 4th molar substrate. The velocity of the reaction catalyzed by enzyme B will be what? Again, simply just draw a generic saturation curve. Here's a generic saturation curve. Where do you find the maximum velocity, Vmax? It says that Vmax is 30. So on this graph, we can put the value of Vmax of 30. There it is, 30. And where do you find the Km? Is found at 1/2 the Vmax. 1/2 of 30 is 15. So this is 15 right here, as shown by the arrow. And then we go to the enzyme line, and straight down, the Km value is 5 times 10 to the minus 4th. So at 5 times 10 to the minus 4th, that is the Km value. And it says in the vignette, in the question, at that value, 5 times 10 to the minus 4th, what is the velocity? The answer is 1/2 of 30 is 15. So those two answers of questions 6 and 7 are very simple, as long as you know what a Km value is. Now question 8, we're coming back to enzyme A. Enzyme A. And answer this question is option C of 20. We're talking more about enzyme A. Now it says at a concentration of 5 times 10 to the minus 4th. No mathematics here. Coming back to this graph at the top of question 6. Let's look at this graph in question 6. On the x-axis, where is the value of 5 times 10 to the minus 4th? Remember, 10 to the minus 4th is 100 times greater than 10 to the minus 6th. So on the x-axis, we're way to the right on the x-axis, meaning we're at the Vmax on that graph. Because we're at the Vmax, that's why the answer is option C, answer of 20.